General procedure for cell culture. This video illustrates the procedure for preparing a cell culture by using living cells of mice, which is a test animal. The cell culture is prepared in 3 phases: isolation of the tissue, disaggregation of tissue, seeding the cultures into culture vessel. Let's see the procedure for the isolation of the tissue. First, a mouse is anesthetized to lose consciousness. Then, its thoracic region is sterilized with 70% alcohol. An incision is done on the skin surface and the lungs are gently removed. The outer epithelial tissue of the lungs is aseptically segregated and immediately transferred into a tea flask containing a balanced salt solution which is stored in a freezer. The second step involves tissue disaggregation. This frozen tissue is transferred into an empty petri dish and minced with a sterile scalpel. The tissue pieces are washed with Hank's balanced salt solution and transferred into another tea flask containing collagenase enzymes. The tea flask is incubated for 4 to 18 hours at 37 degrees centigrade. During incubation, the enzyme digests the extracellular matrix of the tissue by leaving the cells suspended in it. After incubation, the tea flask is taken out and the cell suspension is filtered through a sterile stainless steel mesh to separate the large tissue pieces and the suspension containing free cells. The suspension is transferred into a falcon tube containing HBSS solution and centrifuged for half an hour. After centrifugation, the cells settle at the bottom as pellets and the supernatant is discarded. Again, these pellets of cells are resuspended in the same tube by addition of fetal calf serum culture medium and transferred into sterile trough. The final step is the seeding of cells in a cultured vessel. A 12-well culture plate containing growth culture serum is taken and with the help of multi-channel pipette, a specific amount of suspension is extracted into the first 4 cells. Similarly, the process is repeated for all other wells and the lid is closed. The culture plate is kept inside the incubator for 3 to 4 days at 37 degrees centigrade. During the incubation period, the cells utilize the growth culture serum for growth. The cells divide and adhere to the culture plate. After incubation, these cells can be separated and subcultured for further growth.
For more information, install Learn Pharma app in Google Play Store.